Cheers. Hi, everybody. Welcome to our afternoon workshop. This is Stevie Pederson from the Telethon Children's Institute in Adelaide, and we'll be demonstrating the extraChIPs pack.
Thanks, Tim. Appreciate that. So I started my preparation for this workshop by writing a big session, which is held at this address. And then I realized I'd done too much and there was no way we were going to get through it in 3/4 of an hour. So I then just prepared a little Quarto presentation. So this address here has a workflow if you want to go away and have a play with the package in your own time. But as it is, I think I'll just talk people through what we're going, what essentially is in the workflow. I hope that's okay. There was some data which I have provided, and all the links are in that workflow, because with ChIP-Seq you actually need data, and I have not been smart enough to organise an experiment hub object yet. Eventually I will get to that, but I haven't managed it yet. A little bit of an introduction to me. As Tim said, I'm a postdoctoral bioinformatician at Black Ochre Data Labs, which is part of Telethon Kids Institute in Adelaide, Australia. Adelaide is the traditional lands of the Kaurna Nation, and I'm now working on a project improving health outcomes for Indigenous Australians and some of the conditions which they face. And I'm working on a fairly large multi-omic study. We've got 1,400 participants. We've got genomics, epigenomics, transcriptomics, proteomics, lipidomics, metabolomics, inflammatory markers, ECG scans, retinal scans. I think that's most of it. So there's a team of us working on it. I mainly look at the transcriptomics with a little bit of a foot in the multi-omics camp. I'm also the developer and maintainer of NGS reports. If anyone's used it, if you do use it and you find a bug, then I get really excited when people file bug reports because I just go, someone's using it, yay! So in terms of the history of extraChIPs, prior to working at Telethon Kids Institute, and I'm still working on this project, I was in the Dame Roma Mitchell Cancer Research Labs. And the work that I was doing at that point in time was to integrate multiple chip targets in multiple cell lines. And in particular, we're looking at the regulatory dynamics of what occurs when you activate the androgen receptor with testosterone, because it's been shown that that has an anti-proliferative effect in breast cancer. So we're very interested in what other transcription factors it was playing with, what were the changes to the histone marks. So it was lots of chip targets, and we're repeating it across different cell lines and different breast cancer models with different ER status and PR status and all of those kind of things. To actually do that work, I built a rather large workflow. Well, it felt large, called Gravy. I'm doing a post on that tomorrow. And so this package I really developed as I was building that workflow, and I'd find there were repetitive processes that I was doing, and I built a function for it. So I didn't set out to go, I'm going to build a package. I just realized I'd written enough functions, and I thought, well, let's put this together in a package and just go back and think about it a little bit more carefully. So that's kind of the history of what I've done. And I'm thinking that what I've done actually fills a few gaps between diffbind and Seesaw, which are probably the two dominant ChIP-Seq approaches. I've learned a lot from both of those packages. So really what I wanted to do was just to simplify the processes I was doing in that workflow, and to provide a way of doing differential signal analysis, either using fixed width windows, which is a diffbind approach, or with sliding windows, which is more of a Seesaw approach. And you can do both in extraChIPs. One of the key things I wanted to do was to maintain all the MCOLS information from a GRanges object throughout an analysis. And I wanted to enable this comparison across multiple targets, or perhaps the same target in multiple treatments, and then add a few easy visualization functions. I wanted to play nicely with the tidyverse. I haven't set out to make it a strictly tidy paradigm, written in it with that paradigm, but I wanted it to, at least play nicely. And I'm a big fan of ply ranges, and so it plays nicely with that. And some of the quirks of working with ply ranges and doing multiple joins, resolving those was the motivation for some of the functions. And I invented or created some S4 object classes with the ingest reports package. And I didn't want to do that again. So I wanted to stick with existing object classes because that's what we're all familiar with and what we use. And it was one of the frustrations I had as I learned diffbind is that their object classes do not perhaps play as conventionally with like other ChIP-Seq or other bind conductor packages. And we've since discovered that it's applicable well beyond ChIP-Seq. So some of the stuff I've done here, one of our data sets we're looking at in my current project is DNA methylation. And Sam Buckbury, who's working on that, is very interested in trying to figure out how, well, try to see where there's some connections here. And I've kind of just started looking at AttackSeq as well. So I'm hoping to make the package usable for all of those different contexts. So there's kind of 4 groups of functions that I've written. There's what I call the core infrastructure functions. And you'll see that I've got all these functions with an MC at the end, and they're basically the same as what you'd find in a genomic ranges, but they maintain the MCOLs data. I've got some coercion to AsTibble. So if anyone here is from the tidiness in BioC community, please come and have a chat. I don't want to have been a rogue operator doing something weird off in the corner, but I may have accidentally done that. So that's kind of like some of the core infrastructure. And then I've got my functions for differential signal analysis. One of the things I've introduced is merging windows by the harmonic mean p-value. I don't know if anyone's familiar with the Wilson paper that came out a couple of years ago, so I've implemented that. And just a couple of other strategies which just enhance what happens in Seesaw. Then some nice, well, I think convenient functions for working with peaks and ranges, and then a whole set of visualizations. So that's kind of the layout of the package. So I'll just talk you through a few of the functions and what we can do. So many of you will be familiar with reduce, setdiff, intersect, and union as functions we use all the time when we're working with GRanges objects. And anytime you call these functions, they will drop the information, any information in the MCOLS element. And I want to make sure that that was maintained. So I added these 4 functions just with an MC at the end. So it was fairly easy to figure out what they did. And then they're returning the object as compressed list or atomic vectors where appropriate. And these functions are very much heavily used in all of my downstream functions. So hopefully that's big enough. Perhaps I should have used a larger font. But here is just a really simple GRanges object. We've got 2 overlapping ranges. The first one goes from bases 1 to 10, then 6 to 12. So they overlap in the middle. They've got different IDs, range 1 and range 2, but they both map to the same gene. Now, if you reduce that, that will collapse that to being bases 1 to 12. And that's the classic reduce. You know, it just reduces the overlapping ranges. But then if you do reduce MC, you retain the MCOLS information. So you can see the gene column was simplified, just a nice character vector, whereas the ID was simplified into a compressed character list. And that I've found works fairly well. There is a small computational overhead because there's some stuff happening under the hood, but I found that to be incredibly useful as I've gone through working with the ChIP-Seq data. You can turn off that simplification from the gene column by simplify equals false, but by default it's set to true to try and simplify things, obviously. The Tibble coercion pretty much is very similar to what you might expect, but the range is now placed as a character vector. Instead of having separate columns where you've got seq names, start, end, width, strand, because most of the information is there. And so that was a really simple and straightforward way to do it to my brain. Any more complicated data structures in the MCOLS element are usually, I put them through vectors, vecproxy to turn them back into S3 objects. You can return it in a very similar format to how you would normally get it from as data frame by just setting range as car equals false. And that gives you exactly what you'd normally get from as data frame, but in a Tibble. So you've got both ways of working with it there. And sometimes one is more helpful than the other. To coerce back, I've written a function called a ranges, where you basically move a column to the ranges, And so here we've taken the range, that range column, which is a character vector, just coerced it straight back. You can provide CQ info information, because obviously a character vector won't have that information in it, but you'll probably have it from your original ranges or something anyway. Usually I define a CQ info object at the start of my workflow and just I put that on everything, so I always have everything behaving nicely. One of the other kind of strengths of this call to ranges function is if you've got a GRanges object in your mcols element, you can move that across to the backbone ranges as well. So here I've just added like the peak centers. These are all pretend, obviously, but the peak centers in a column called center, they're just single nucleotides, and then use call to ranges, and now they've moved across to be the backbone ranges. So by switching, you can easily manipulate ranges, particularly in the case of a fixed width analysis, where you've perhaps got all of your peaks loaded, but then you want to fix the same width and center them. You can easily just switch things backwards and forwards between the backbone and your mcols. So they're kind of like the core infrastructure functions which I've introduced. And in terms of working with peaks, the larger workflow I've got, there were two analyses in there, which is why I was a bit silly. One of them is for the estrogen receptor, and the other one is for the histone mark H3K27AC, and they're both under E2 as control and E2 with DHT, so estrogen, and then estrogen with testosterone as the treatment level. So that's the demonstration data I put together, and this is just me preparing the metadata, and the metadata on the left is actually what I would have. So I've got basically a samples data frame for each. So you can see we've got 3 E2, 3 with DHT, and it's actually taken just from SRA, and all the identifiers are there. And I've pre-processed this to just give chromosome 10. So if you work on this at home, it's really manageable on a laptop. But obviously, doing sliding windows for a whole genome is not so viable on a laptop. So the data that I've provided in that workflow, there are 3 folders. There's annotations, and then for the estrogen receptor, there's bigwig files summarized by treatment, and then narrow peak and BAM files. Well, what happened? Okay. And then for the histone mark, I didn't put the narrow peaks in because I wanted to use that to demonstrate sliding window analysis. So you don't need peaks so much for that. So that's what the data would look like. As I said, I like to create things at the beginning of the objects, at the beginning of a workflow. So I've just shown you what I've done here. There's a CQ info object. There's a GTF here, which I've split by feature type. So it'll have a separate element for the genes, the transcript for exons. I've downloaded the blacklist, the ENCODE blacklist, and I've found the grey list from my input sample. I've just put them there in a bed file. And I set up colors to make sure that every time I make a plot, I've got the same colors representing the same thing. I like to do that at the start of my workflows. It just makes communication much easier when you're always doing the same colors and the same plots. Okay, so when we're working with peaks, often we'll have run Max2 or some other program to identify the peaks where there's a real bump in the signal for a particular chip target. And they're often outputted in narrow peak format. So that's what I'm used to working with. There's also broad peak, and the package can work with them nicely. And what you'll often do is call peaks across individual replicates and then merge them to find like consensus peaks for a treatment. And so that's kind of the first step that I would do in an analysis. Usually you'll merge the peaks across a treatment group, and then you kind of work out how to combine them across different treatments. Okay, so the function that I use for doing that is I use import peaks. Now, of course, we can all use import bed or whatever, but this always gives you a GRanges list because they're always going to have the same structure, and you're probably going to turn it into a GRanges list, so just do it from the word go. When you provide a blacklist here, they're automatically emitted during import. When you provide a CQ info object here, only sequences held within the seq-info object are imported. So it does a lot of the filtering for you during the import stage. If you're dealing with narrow peak files, the actual peak center in coordinates, not in peak, is calculated as well. So you've actually got the centers to work with from the word go, which I find really convenient for me. So here's how I would import my peaks. Just import peaks. I've got my file list there. Passed it my black and gray list, the seq info, and I said, give me the center. And then this is kind of what you get. There's my GRanges list. I've got 6 of them, and the standard narrow peak columns. Those who have worked with narrow peaks before will be familiar with that. But the center column is the extra bonus column that I'm giving you there. So when you want to then see what your replicates look like, because we've just imported six narrow peak files, just plot overlaps will give you a straightforward look at how they line up against each other. So I've just literally called the GRanges list, set plot overlaps, and I've set the set columns to be one of those vectors I defined earlier. So we know which ones are estrogen ones, which are the blue, and DHT treatments in red. Good way of just spotting any outlier samples. The next step, once we've kind of had a look at our replicates and we're happy that everything's doing the right thing, we will want to make consensus peaks here. So just the simple function, make consensus. Now here I've just passed it the first 3 samples because they would be E2 or the control. And when you call make consensus, I've added var equals center. And so I'm keeping all of the information in that center column as that compressed list object there. And I think you can see what we might want to do with that a little bit later down the track. And then I've also got some logical columns saying which replicate the actual peak was found in and how many replicates. I've also applied P equals 2 over 3. So make sure the peak, only return the peaks found in 2 out of 3 samples. Yeah.
So how are you actually making the consensus peaks? So like, what's the algorithm? Okay.
Here's one I prepared earlier. Thanks. The default is to just have the union of ranges. So here's like 3 kind of overlapping ranges, which I've made up. And so by default, you would do the union. But if you set method equals coverage, you can return only the region covered by 2 out of the 3 replicates. Yeah.
What happens if you have like 2 peaks in one sample overlapping 1 peak in another?
Sorry, I didn't actually quite hear that. Is there a mic that we can take around or something?
I can yell.
Yeah, okay. Oh, yeah, okay. So yell, and I'll repeat the question so that people online can hear.
Yeah, so what happens when you have multiple peaks in one sample overlapping 1 peak on another sample?
Yeah, that's a great question. The question was, what happens if you have multiple peaks in one sample overlapping 1 peak in another? So if you use method equals coverage and you said, I guess you set it to be both samples, you could get just those slices where they overlap. Or if you use the default of method equals union, it would be the entire section. Every time you try to imagine one of these scenarios, you can always think of a million exceptions. It's like, I'll handle that scenario. And then, of course, something else happens, because you get weird double peaks appearing sometimes as well. Great. We've got a mic. Thanks, Tim. Okay, so you can make consensus peaks either way, just by the union or by the overlapping slices. Of course, this has flaws as well, because you can imagine you can get some tiny little peaks. So, and you can actually set in make consensus the minimum width. So you can throw away anything less than 50 base pairs or whatever you choose. So here's how I would go about things in a proper analysis. I'll probably code it a little bit more confusingly for myself, but here I've just created a list of E2 and E2GHT. I've done an lapply here saying, give me just the center column back, and then make it a GRanges list. And here's what that looks like. So here I've got like the consensus peaks for the estrogen samples and then the consensus peaks for the DHT samples. And then I can try plot overlaps again, and I get a Venn diagram this time. You'll always get a Venn diagram when n equals 2, always get an upset plot for n is greater than 2. Yeah, you can't do a Venn for less than, for more than 2, but you can do an upset with 2 if you want. But so now I've made those treatment-specific peaks, consensus peaks. I can just do make consensus again, but this time not applying any threshold to get kind of like the union peaks, which is a peak in anything, and I've automatically added the center. Well, automatically I wrote the code, but I've added the center here. And so when I'm about to start my fixed width analysis, I have the peak centers already sorted out and propagated through the analysis. Okay. Once I've kind of got these peaks all sorted, one of the things we want to do is find what regulatory regions they map to, what genes are they likely to be doing things, are they likely to be regulating. That's really the point of it, not just going, hey, there's a peak, let's go have lunch. We want to actually answer the proper questions. So one of the functions I've included is define regions. So if you pass it a set of ranges which represent genes, transcripts, and exons, it will return a GRanges list of unique non-overlapping regions representing promoters, introns, upstream promoters, exons, and so on. There's another actually, sorry, I've jumped ahead. And map by feature is my way of assigning peaks to the most likely genes. And the way I've done that is to incorporate promoter information, enhancer information, and long-range high-C interactions if you have them as well. So trying to get, the best mappings I can. So here's define regions, where you would pass it ranges representing genes, transcripts, and exons. Tell it how far you want where you want your promoter to be, or what you consider to be an upstream promoter. Do you want introns actually annotated? Because I don't get why people are so interested in a ChIP-Seq binding to a sequence that goes from an intron. That's an RNA issue, not a DNA issue. But anyway, people like it. Or introns and exons, it's just gene body, I say. Proximal, intergenic, and then distal intergenic are the other things. And so these regions are defined hierarchically. So if you've got like 2 transcripts where your promoter regions kind of overlap, that's just merged into a big promoter that can be working with either transcript. So it just does it in that hierarchical order. And here's what you would get just off that little subset of chromosome 10. So you've kind of got the promoters. I've just given you the promoters here. Then what genes they map to, and what transcripts map to as well. So then we can use that to then map our union peaks using best overlap. Again, you can pass a GRanges list or a GRanges object and just tell it what column you want to grab the information from. And so there we go. We've got our union peaks mapped to whatever regions they seem to map to. Hopefully that's fairly easy to follow. And now we've got those peaks mapped. I've written a function PlotPi. So we can just do a nice little pie chart there showing the distribution. You can scale these slices by the width of the peak as well if you really want to, or you can just do them by counts. One of the things which I actually really, I'm pretty happy that I thought of when I was writing this function is I've used the glue syntax for the labels. So you can actually really customize those labels for PlotPi with a lot of detail. So here's what I've done. So here I've called string wrap just because I wanted to format what was in the column I'm filling by, put a couple of line breaks. The totals are available as N, the proportions are available as P. And so you can really customize these labels, and you can just obviously put plain text, you call them all Bob if you want. But yeah, so that's a really nice, I found that incredibly useful because you make the default plot and you go, oh, they overlap, I'll just tidy this up. And being able to really customize them is nice. So map by feature is another way another one of mine. So here, if we take our ranges and then we pass it genes that we want to map to, but then regions we've annotated as a promoter, regions we've annotated as an enhancer, or anything with a G interactions object for high C data or any other conformational data. And then ranges will be mapped again hierarchically. If it overlaps a promoter, we just map it to that promoter. Although, if there's a high C interaction, it will do that too. If it overlaps an enhancer, it'll map to all genes within a given distance. If it maps to a long-range interaction, both genes at either end or any gene at either end, and you can set how far away you want the peak to be to either end or the gene to be. If you haven't mapped it at that point, it'll map to all overlapping genes or the nearest gene within a specified distance. So I found that was the best way for me to map peaks to genes. And so here I am adding them to union peaks. We've got the union peaks, the centers, the region they map to, the gene IDs that they map to as well. So hopefully we've got all the useful information we need about our peaks there, and hopefully it's as accurate as we can be. It's always challenging because genomes never behave. So moving into differential signal analysis, as I said, I've enabled two approaches. There's a fixed width analysis, very comparable to diffbind. The idea is you would bring in a set of bring in your peaks, define your set of fixed width ranges, you'd center them how I've shown, resize the fixed width. Then usually I just read in the counts using Seesaw, and that gives you a range summarized experiment, and I pass that to fit assay diff as the function I use for differential signal analysis. Sliding windows are very similar to Seesaw, where you basically define your window size and step. I've added a new function for filtering those called dual filter, where you provide a set of guide ranges. Say, we expect to see signal here, so give me back the peaks that look something like this, or the windows that look something like that. And then you pass that to fit assay diff and merge neighboring windows for your final results. When you've got your range summarized experiment object, I've added a few functions. I think I may have reinvented a couple of wheels here, where we just plot assay PCA or RLE or densities. And then when you call fit assay diff, I'm passing everything to GLM QLF within EdgeR, or you can pass it to limit trend. I know don't think a lot of people use limit trend for ChIP-Seq analysis, but I found it works really nicely actually. And then when you've got sliding window analysis, you merge by the harmonic mean, or merge by col, merge by sig, just wrap Seesaw methods, but they do a lot of the mapping and overlapping, all self-contained, and they all give you back the key range. So in the case of Seesaw, you might choose the highest signal or the lowest p-value or whatever, and so you get the range that came from returned as well. I find that useful. Okay, so fixed width analysis. That's just all the code. I've now loaded everything in. Plot assay densities just using ggplot. So it's fairly easy to work with. And plot assay PCA, again, you just choose the assay, transform how you need, and pass the colors. So everything is fairly straightforward with those. When it comes to the RLE plots, you can do them in a really standard manner. Just plot assay RLE, tell it which assay to plot, and it will do it. Or you can group them by treatment. I was actually working with, say, the antigen receptor is a classic for this, where it's cytoplasmic until activated, and then it's nuclear. And so you get vast differences in signal. You can actually do RLE by sample, taking the means within treatment groups as well, if you'd like. We also had another transcription factor that we knew was nuclear, but we also thought the antigen receptor brought more of it when it was activated. So it wasn't a strict binary, none to 100 type scenario. So the RLEs, there's actually a bit of nuance you can really play with in the package. Again, sliding window analysis, here's all my code. When you just do it on the data set I've given you, there's 467,000 windows. And then I would apply my function dual filter, where basically I'm saying, here's the ranges, and I just they're called by max2. And that filters it down to to 1900 windows, at which point in time you can do the analysis. And when you call dual filter, it adds a log CPM assay as well, which just makes plotting easier. And if you do want to use limit trend or do any other additional steps, I was actually using smooth quantile normalization in the larger workflow, and that made life easy for me. So here's my differential signal analysis function. I call it fit assay diff. It's deceptively easy, but you can actually do a lot with it. So the two methods I've implemented, like I said, QLF, which is the GLM QLF from EdgeR, or LT is short for limit trend. If you're fitting log CPM values, you can apply the range-based null hypothesis from TREAT, if you're familiar with the TREAT approaches here. I noticed DiffBind has actually just put that in the last bioconductor release as well. You can normalize within treatment groups, because I was dealing with that androgen receptor situation and the other one that may have been piggybacking in. So if you're normalizing, say, by TMM, you can set it to be normalized within groups. If you choose library sizes default to the actual complete library size, but you can also set it to be whole sum. So there's lots of options for playing with things there. If you want to normalize by using offsets, you can pass the offset there as well. So you can actually do quite a lot with this one single function, but the defaults essentially are library size normalization and EdgeR fits, which is pretty much the DiffBind defaults. So when you do fixed width analysis, there's my code. The final bit at the end, actually, I'll go back. This is the bit that's worth looking at. So here, I've just called my estrogen receptor summarized experiment, define my design matrix. I'm normalizing using TMM normalization. I'm saying return it as ranges, not as a summarized experiment object. And I'm only interested in peaks that are showing about a 20% change in signal. So FC equals 1.2. And then add diff status. I'm just adding the status of each peak there. And then to print the results, because they look messy, I've just done a quick tibble conversion. And this is the results, basically how it looks. Of course, once you've done that, I've added a function called Splot plot split donut, where you can then say, okay, well, what regions were all the changes in? Of course, we've only got 188 peaks in my toy data set here. But again, I've used the glue syntax for these labels. So you can do a lot with customizing these labels. As you might be able to tell, on these outer labels, I've actually added what the inner region is as well, because you can do that using glue. And as far as I'm aware, hopefully I'm right on this. I think it's the only function where you can set your inner and outer color palette separately. I haven't seen any other donut functions do that. So you can see I've got the inner palette and the outer palette set separately. So again, I've found that one really fun to work with, because let's face it, these donut plots are fun. I can explode segments using just a regular expression query. So here I was querying for the pattern increased, and all the ones that matched increased were exploded. And you can set a p-value range, or a proportion range, sorry. So, show me the labels for everything between representing 5% of the ranges, or 10% of the ranges, or you can just move that around wherever you want. Here I've set it, anything less than 5%. Okay. I'm just keeping an eye on the clock. So sliding window analysis, it's basically the same approach, but here I'm merging by the harmonic mean P, and I'm throwing away where there was only one window, because they're usually low signal regions. I'm saying merge within window size +1, because sometimes you get these weird little things happening, particularly with histone marks, where you've got that trough in the middle of them. And actually here, the key value range, I would take as the merge region. There's a lot more in the manual which will explain this. But for merging by HMP, I'm returning a weighted average of the log fold change, a weighted average of the expression, and the P, because a harmonic mean P is actually a weighted average, so I'm doing the same. Well, it's an average at least. I'm using the average, the weights, which are the inverses of the P's. And then add differential expression status, and then this is what you get. Fairly standard looking results. With sliding windows, you tend to map the final ranges to the genomic regions or to the genes afterwards, because you're dealing with windows. And so I've done that here. It's the same process, just done after the analysis, not before the analysis. And now we're getting to some of the other fun bits that I really like to do, is comparing the results. So when you're comparing two targets, you might want to see where one's bound and the other one's not, where they're both bound, they're both moving together, one's moving, the other one's not, all of those different things. So I've written map GRL cols for that process. Plot, split, donut, obviously you can do whatever you want. There's a million ways you combine the data. Plot pairwise, if you want to show what the signal is like, where both are present or only one's present, show where they're moving together. And then plot HFGC is my little mini genome browser plotting function, which I think I will get to. I've got 5 minutes, and if I talk fast enough, I'll get there, but no one will understand what I'm saying. Okay. So when you're comparing two targets, I'm working with GRanges List objects all the time. And so you're going to need the same column names, and you get slightly different output from merging by HMP and from just doing a standard fixed window analysis. So here I am just setting a GRanges List object, but I've just done a little bit of a messing around with the histone data, which is what I did the sliding windows on. And so that just gives me the same columns in both results. So here's all results. Oh, no, I didn't show you all results. I should have. Anyway, so it's basically a GRanges list. They've both got the same column names. And when you do map GRL cols, you can do this with a GRanges list of any size. Basically, I'm saying now map the log FC column and the status columns and collapse the genes where they're the same. And so you can see here the first range was only found in the histone data, wasn't found in the estrogen receptor data, and it was unchanged. But then the next range, we had values for both, and we know what genes they're mapped to. So it's a really nice, easy way of mapping, multiple elements of a GRanges list and going, where do they work together? And you can choose any number of columns, any number of GRanges list doesn't have to be of a particular size. And so you can do some really nice, easy, straightforward things here. So here I've just mapped the log fold change in the status columns and the gene name. And I've said, give me the results where it's either increased or decreased in both of the status columns. And so there we go. There's our three ranges where both estrogen receptor and the histone marks are increasing. And we know what genes are mapped to straight away. Plot pairwise only works with the GRanges list of 2, or you can have a GRanges list and tell it which 2 you want. But here we're comparing the log CPM values, and I've got these little box plots on the side. So you can see where they're moving, where they're both found, but you can't have a point on the plot if you've only found it in one. So that's why I've added these box plots at the top. So you can see at the top, this is the estrogen receptor signal when only ER is present or when both are detected. And then on the other side is the distribution of histone signal when both are detected or without estrogen receptor. Probably more informative when you've got more points. We only have 188 here. Then if you want to do plot pairwise on your fold change, again, it's calling this map GRL cols under the hood. And it automatically, well, if you ask it to, it will provide you the gene names for the most extreme point in each group, and it does the collapsing. Do I have a mouse that I can show you? It collapses groups saying both unchanged or both increased, and colors by those groups. And again, you can see what the fold change distributions are like. So here's how ER looks when there's no histone mark. Here's what ER looks like when the histone marks are also increasing. Here's what ER looks like when the histones are decreasing, and so on. So you get nice summarizations of how everything is working together. And that's I haven't tweaked that plot. That's just the code. I've added some scales because they're just ggplot objects. Okay, finally, in my 1 minute. Plot HFGC is one of actually the first functions I put in the package, and it's for plotting high C features, G models, and coverage, because that's the kind of thing I was dealing with in the gravy workflow. And reality is, once you've defined, your high C object, what features you want, what the G models are, and what the coverage tracks are as bigwig file lists, you just go, all right, here's the range I want to look at, bang. Here's the range I want to look at, bang. And you can just go through multiple ranges and get the same kind of results. All of the tracks are optional. Obviously, won't be very informative if you have no tracks, but you need to think about these things when you're writing an R function. I'll quickly just, there's a lot of flexibility in how these tracks work. For the coverage, I'm expecting to see a bigwig file list. If you've got one bigwig file list, it will split out all the files onto separate tracks. But if you pass a list of bigwig file lists, each list element will become a separate track with the coverage overlaid on top of each other. And so that's what I've got here, where I've got the summarized bigwig files for the estrogen receptor with the 2 treatment groups. So I passed a list of bigwig file lists here, and then the histone marks are on the bottom. You can annotate using the actual results from your analysis. So I've got little red bars saying it's increased above each of those. There are the gene models and those features I defined earlier. There was no high C data for this particular data set, but it plots fine if you have high C interactions as well. So I find that really, really nice if I go, all right, give me the top 10, just set up a loop and just spit out the plots, see what they all look like, or do something along those lines. So yeah, that's the function kind of in most of it, and I finished on time, and I probably spoke too fast. Sorry about that if you couldn't understand my accent, but hopefully it's useful. I'm using it all the time, and I hope other people find it useful as well. Any questions? Oh, lots.
Sorry, I stepped in a couple of minutes late, so sorry for you to cover this. But I know you designed this package primarily for ChIP-Seq, but have you used it for TAC-Seq, and are there any adaptations that you would suggest for that mode of data?
Currently working on adapting it for methylation. I haven't got ChIP-Seq data right in front of me, but as I'm finishing up the gravy workflow, I want to put a TAC-Seq into that, and I've just been going back and tailoring a few functions to be able to handle, because with a TAC you don't have the input sample, for example, as a thing. So it should work with a TAC-Seq. This dual filter function for sliding windows should work with a TAC-Seq now as of the latest version, but I haven't actually got a TAC-Seq data in my hot little hands to test it on, and that's very high on my priority list to actually test and see how it performs.
Hello, thank you for the great talk. I'm kind of curious on your last bit on the plot HGC. In your high C data, can we see the interaction matrix, or how does it plot?
Yeah, the way I'm plotting it is I'm really wanting the high C data to be restricted to significant interactions.
So there's a
but
threshold.
if you just put a whole interaction matrix in there, it's just going to fritz out. So usually it's a G interactions object where you've just got interaction tail 1 and or anchor 1 and anchor 2. And I'm expecting that you will have done some filtering on that beforehand to just retain informative interactions. If you put them all in, yeah, it'll work, but it probably won't be that informative.
I see. Thank you.
More questions? Well, like you said, you're right on time. Thank you so much for a wonderful talk.
No worries. Thank you.